Okay, we're going to pull up the next set of slides, and I have the great honor of introducing someone who doesn't need to be introduced, Dr. James Holland, who will be the next speaker. As you can see, this plenary session of the last meeting of CALGB, as we know it, has been designed as this latter part of it as one of insight and reminiscence from the previous group chairs. And the first one to provide that for us will be Dr. Holland.
Just that. Okay, good. Laser pointer. Thank you. Very good. I'm going to give you a little history of the CALGB. Not at the urologic level. I hope bathroom break won't be necessary. In 1953, the clinical center opened, and I was there the day it opened, and was privileged to meet some of the scientists and to work with them at the NCI. I made $7,200 a year, which was a major increase over what I had been paid. But Roswell Park offered me a job for $11,400, and I left in November '54. Shortly before I left, Gordon Zubrod was appointed the clinical director, and he said, "I don't have a program in acute leukemia. Would you mind if I continued your protocol?" An offer which had never been comparable in any of my past experience. And shortly after that, he recruited Tom Fry from St. Louis and called me back from Buffalo, which was an overnight train trip in those days. And Tom Fry and I met each other and agreed, and he then carried on, and agreed to collaborate and cooperate. And I recruited George Selkirk at the Children's Hospital in Buffalo, and the first study that we did was a combination chemotherapy study, which was based on the work of Lloyd Law. Tom Fry and I became joined at the hip, and as we published Cancer Medicine with our colleagues and have continued in contact, and of course he regrets he can't be here, and as many of you know, he has Parkinson's disease, and his flesh is weak, but his spirit is willing and strong. And I spoke to him last night on the phone, and he speaks slow, as people with Parkinson's disease do. So he put his message in an email which arrived this morning. And I'm going to read you the email from Tom Fry. Greetings and salutations to my colleagues on the eve of CALGB's new formation. When I think back to the humble beginnings of this organization in 1955, I'm amazed at what it has become and the crucial research that it has fostered. Driven by advancements in basic science, the ultimate clinical goal of curing cancer has been achieved in substantial numbers of patients with leukemia, lymphoma, breast cancer, colon cancer, and osteosarcoma, to name a few. As CALGB enters the next phase of its development by merging with the North Central Group and the American College of Surgeons Oncology Group to become a new and more dynamic organization, we can take pride in past achievements and a future of even greater promise. I am confident that the same energy and intellectual sophistication that has led to our current level of success will lead to ever greater scientific conquests. With my best regards, Tom Fry. I certainly endorse every aspect of that. Now, when Mary Lasker, who was the admiral of the American Cancer Revolution and planned most of it, arranged that the Cancer Chemotherapy National Service Center be established, it had 4 panels: a clinical panel, a screening panel, a pharmacology panel, and a drug discovery chemical panel, as I recall. And the clinical panel was chaired by Dr. I.S. Ravden, who had been Eisenhower's surgeon. Eisenhower had, I guess, Crohn's disease or some intestinal problems, and Ravden operated on him. And he said, I don't know anything about chemotherapy, but I know how to knock men's heads together. And he had been a general in the Army, and I'm sure that he had knocked some heads together, but he organized the clinical panel. And its most influential member was Gordon Zubrod, who recognized that the British had undertaken collaborative clinical trials in tuberculosis and had proved that para-aminosalicylic acid was useful in addition to streptomycin. And he suggested that, Zubrod suggested that we have groups of investigators who would undertake clinical trials using scientific methodology which had been laid out by Gadam, a Scottish pharmacologist. And there was an acute leukemia group A, headed by Joe Birchinall, who was senior to Tom Fry and me, and had been working leukemia for a long time, and was enormously generous in sharing his data. And we became acute leukemia group B. Acute leukemia group A morphed into the Children's Cancer Study Group, but we were proud of the name CALGB and kept it until many years later when, as I will point out, we picked up a lot of information from leukemia and applied it to other kinds of cancer, and so added it up front to CALGB in the middle 70s. So this is a picture of acute leukemia in childhood. There were no pediatric oncologists at the time, and neither Tom Fry nor Jay Fryreich nor I were pediatricians, nor had we trained in hematology. But leukemia infiltrates organs and makes them into tumors and creates clinical abnormalities that one can recognize: a normal mouse on the left, a leukemic mouse in the center, and a near-dead mouse on the right. And by the time this happened in 1953, when I went to the Cancer Institute, 2 other major observations had been made. Ludwig Gross had shown that these leukemic mice, AKR leukemic mice, it was caused by a virus. And nobody has exploited that as effectively as needs to be done, and that's still in the future. And Lloyd Law had shown in a transplanted leukemia related to carcinogen administration that the tumor cells were resistant. There were resistant members of the population from the very beginning, which he did using a method similar to Luria-Dilbeco randomization techniques, in which multiple colonies of the original tumor were shown in successive generations to have different sensitivity. And he also showed, as demonstrated here, that combination chemotherapy was useful. And indeed, he used azaguanine, which was a purine analog that antedated 6-mercaptopurine. And here, a combination of methotrexate and azaguanine in homozygous mice, DBA2, or in BDF1, demonstrating that, in fact, they were more effective as treatment in animals that had a genetic component able to resist it, since the leukemia had arisen in DBA2. And in the middle, methotrexate first, and sequentially, a purine analog, and on the right, the combination given simultaneously. This information was available, and when I went to the Cancer Institute, I was assigned to work with Lloyd Law, who was going to give a scientific presentation to the scientific directors of the NCI. And I learned a lot, and he and I became lifelong friends. He died a couple of years ago at 91. So I put together 6-mercaptopurine and methotrexate and began to treat adults and children, and that's the protocol that Gordon Zubrod wanted to continue. And that's the protocol we published. It's the first publication of a collaborative study in the United States, and there was no difference in the two arms we chose, one of which was 6-MP and methotrexate given daily, and the other was 6-MP and methotrexate given every third day, which was, in fact, the demonstration that Abraham Golden had made, that you could give more methotrexate when you interrupted the dose. So there was no difference in the two arms, and we went back and restructured a subsequent study looking at sequential administration. And then Jay Freireich led a study that's the basis for a great deal of what we do in cancer research today. If we induced remission with steroid and let children be randomized either to no treatment or to 6-mercaptopurine, you could demonstrate that 6-mercaptopurine had an effect on the tumor when the tumor wasn't visible. And those patients treated with 6-MP indeed had a beneficial effect, and that is the precursor for the demonstration of adjuvant chemotherapy. This study, which is study number 7, made use of the observation originally demonstrated at the Cancer Institute in a sequential study, and this was the first demonstration in a group study that vincristine and prednisone together were a good induction treatment. And then children were randomized to either methotrexate intermittently or by mouth on the daily basis, which is what Farber and Birchnell had been using. And the outcome of it was the first real change in survival, with this being the intermittent higher dose methotrexate, and this being the low daily administration. And of course, when children got out here, this is 4 years, when they got out at this level, we began to see the side effects of chemotherapy, which had not previously been recognized.